Hi there everybody, my name is Jayton Cranmore and welcome to another week of Anatomy at SI. Today we're going to look at Laboratory 9, which is going to encompass all of our special senses. First, let's take a look at the general senses, which is section 1. Let's talk about the 3 different types of receptors that we're going to encounter. So, first of all, there are exteroreceptors, right? EXT, prefix meaning out, right? EXT, exterior. Exteroreceptors are going to receive signals from the outside, right? Interoreceptors, those are going to receive things from the inside, right? Interior, intero. Interoreceptors receive signals from the inside. And then proprioreceptors, these are receptors that are found within our joints, right? They help us maintain balance. And like when my eyes are closed, I know where my arm is in space, right? There are a couple different types of receptors that help me do that, but proprioception is the main way that's done, right? Proprioception is kind of how we orient and understand where our body is in space, right? Especially with balance. All right, let's go ahead and talk through the different receptor classifications now. So first, we've got chemoreceptors, right? Chemoreceptors detect chemicals within our internum, right? Notice I said internal, right? So what are chemoreceptors likely going to be? Interoreceptors, right? Yes, most of our chemoreceptors, if not all of them, are going to be chemoreceptors. And so those chemoreceptors detect chemicals like maybe in our blood or in the lymph tissue or in maybe in different areas where there's chemicals that we need to, monitor. What about thermoreceptors? Thermo, prefix meaning temperature, right? So yes, thermoreceptors are going to receive temperature signals and kind of make sense of those, right? Next, we've got photoreceptors. Those are the rods and cones found within our eye. They detect light, right? Cones are going to detect color, right? Color, cones. I like that the C's go together. It makes it easy, alliteration. And then we've also got rods, right? Rods are going to detect black and white light, also help us with gray. So like when it's dark outside and you can kind of see shapes, but you can't really make out color or what things are, that's your rods kind of kicking in to help you still be able to see and make out where you're going. But cones are what allow us to actually see sharp visual color, right? Okay, next we've got mechanoreceptors. Mechanoreceptors are going to help us sense touch and pressure. These are both exterior receptors and interior receptors, right? We have these pressure receptors inside of our body as well to kind of help us maintain like different areas where there might be higher or lower concentrations of pressure, like maybe in our stomach or in our intestines or maybe in other areas, right? Okay, and mechanoreceptors are more with mechanical touch, mechanical pressure, right? If there's too much physical force being pushed against something. And baroreceptors have to do with fluid pressure, right? Baroreceptors, when I say fluid, remember that fluid can be either liquids or gases, right? A fluid can be either a liquid or a gas, and so baroreceptors are just detecting that fluid pressure, like primarily in our arteries, right? We have baroreceptors in our aorta, and also up in our carotid that help us kind of detect how much pressure is going both into our overall body and then also up into our brain, right? And so with that, the last one we've got is nociceptors. Nociceptors, no is in the name, right? No one likes pain. Nociceptors, no pain, right? Nociceptors detect pain. I love that because I don't like pain, right? I always say no to pain. Nociceptors detect pain. All right, next we've got the gustation taste buds, right? I don't have a model for these ones, but I'm going to go ahead and talk about the different papillae and kind of where they're found, and some of them have special functions that we'll talk about as well. Okay. First of all, the associated cranial nerves that help with gustation are going to be the facial nerve and the glossopharyngeal nerve. The facial nerve is going to innervate the first anterior 2/3 of our tongue, right? The anterior 2/3, right? The first 2/3 of our tongue. Next, we've got the glossopharyngeal nerve, which is going to innervate all of our taste sensation on the posterior 1/3 of our tongue. I try to show you that, but that'd be kind of awkward. All right. Let's go ahead and talk about the Vallee papillae, right? Again, I don't have a model with me, but if I did, I would point to the back of the tongue, right? Let's say that's the tongue right there, right? We're free-handed a model for you. If that's the tongue, we've got Vallee papillae right at the back here, right? They're like these little bumps that kind of sit at the back of the mouth. And those Vallee papillae are going to detect bitter taste and kind of form this V shape at the back of the tongue. They're pretty easy to see. All right, fungiform papillae are going to be around the tip of the tongue, so they're going to be kind of around this area, right? And those are going to, and yeah, they're just kind of general taste buds. They're helping with like that umami taste. Umami is meat taste, right? And then also some bitter and sour taste, but not as much sour, right? This is mostly going to be sweet and umami tasting. We've also got filiform papillae. Filiform papillae are going to feel pressure, and they're found all over the tongue, right? And around the entire tongue, our whole tongue can sense and feel pressure. Then we have the foliate papillae, which are going to be found kind of on the sides of the tongue there. And they kind of look like gills on the side of the tongue. But what they do is super interesting. They actually detect sour taste, right? And so that's their primary function. And I always remember this because I don't know if everybody's had the same experience, but I used to love warheads when I was a kid. And I still will partake in them every now and then, but not always. And so when I would eat a warhead, or even sometimes when I'd see a warhead, the sides of my tongue and like the sides of my mouth would just start going, right? Like it was this really strong sensation. It's hard to describe, but it was like this weird, sour, strong, like almost preparing for those warheads, right? And looking back, that was actually those papillae preparing for that sour taste. And when I would pop the sour candy in my mouth, right? Those papillae down there, right? The foliate papillae are the ones that sense that sour taste, and they just get overloaded. If you ever run a warhead back down on the bottom of sides of your tongue, you will have a very not fun experience. Unless you like sour, then it'll be kind of a roller coaster for you. But anyway, gustatory cells, those are going to be the cells that sit beneath all of these different papillae, right? They're kind of associated with these papillae in that they transport the signal down and into its various sensory area, right, where it's supposed to go. In the insula, I believe, is where the gustatory sensory center is. But with that, let's go ahead and talk about the olfactory tract, and yeah, the whole olfactory receptor system. For that, I'm going to have to grab a model, so I'll catch you in just a sec. All right, so I've got the nose for us here. Let's go ahead and get started with the olfactory receptor system. So first, let's talk about the associated cranial nerves. So there's only one cranial nerve associated with smell, and it's the olfactory nerve, right? That's the nerve that's going to extend off of the back of the nose and up and into the brain. And the way that, at least in my lab manual now, it's kind of flipped where it starts with the olfactory tract and goes to the hairs. I want to start with the hairs because that's where smell signals are actually like starting, and it'll travel up and then into that nerve if we travel from this direction. So I'm starting with the olfactory hairs. The olfactory hairs are going to be these chemoreceptors that are found kind of at so first of all, we've got the nares here, right? The nares is the nostril. If we travel up through there, we're going to go up into our nasal cavity, right? We've got the nasal conchae right here that are helping to circulate air and warm it as it travels into our windpipe. We're not too concerned about that right now. Right now we're traveling even further up, and then we meet these hairs, right? And these different hairs, if they don't look like this, of course, in the nose, they're much smaller. But these hairs are going to be little chemoreceptors that kind of capture the chemicals that are found in the air and then transport them up and into the olfactory receptor. The olfactory receptor is going to be a little cell that's directly beneath the that's connected to those olfactory hairs. And that receptor is going to kind of kick on a signal saying, hey, we've received a smell signal. That olfactory receptor then sends its signal up through the receptor and into these different nerve fibers. These nerve fibers are going to transport that signal even further and up and into the olfactory bulb. The olfactory bulb is found on top right here. This is a huge huge bundle of ganglions, right? Remember, ganglions are just an area of lots of cell bodies, right? And this ganglion of interneurons are going to help transport smell signals from these nerve fibers up and into the olfactory tract. The olfactory tract is going to be this region right here. It's the part of the olfactory nerve that extends directly off of the olfactory bulb. This is the olfactory bulb, and there's the olfactory tract, right? So running through that one more time, we've got the olfactory hairs down here catching those chemo signals, transporting them into the olfactory receptor, right? Olfactory receptor is going to send its signal through the nerve fibers and up and into that olfactory bulb. That's that big old root ganglion right up here, right? Now we're going to kick it back off into the olfactory tract, which is just an extension off of the back of the olfactory bulb that carries signals into the olfactory nerve, sending it up and into the olfactory brain center, right? The olfactory cortex. All right, so that's all of olfaction. I'm gonna go ahead and get the eye model. I'll catch you right back in just a sec. All right, let's go ahead and talk about the next set of sensory structures that we want to know, which is the eye, right? The eye is extreme. There's tons of stuff going on in here, but let's go ahead and talk about it, right? The first thing that you need to know is that there's one associated cranial nerve with sensory innervation in the and on our eye, right? That's the optic nerve. The optic nerve is going to synapse on the back of the eye and transport sensory signals back through the thalamus and into the occipital lobe, right? Let's go ahead and talk about some of the different areas within our eye now. So, the first one is the anterior cavity. The anterior cavity is going to stretch from the cornea, which is on the outside. That's part of the fibrous tunic. It's a clear form or clear bit of fiber, right? That kind of covers the opening of our eye. So the anterior cavity extends from the cornea all the way back to the lens, right? And it is divided into two separate chambers. So we've got the anterior chamber and the posterior chambers of the anterior cavity. The anterior chamber is going to be found in front of the iris, whereas the posterior chamber is going to be found behind the iris and between the iris and the lens. That's the posterior chamber of the anterior cavity. Now, we've talked about the anterior cavity and its two chambers. Let's go ahead and remember, the two chambers are divided by that iris, right? That's a very important part, or important aspect of the eye that you need to remember. Okay, next, let's talk about the posterior cavity. The posterior cavity is going to be found kind of behind the lens, right? The lens is this structure right here. We'll talk about it in a little bit as well. But all of this space back here behind the lens, right? That's the entire posterior cavity, and if the anterior cavity is filled with aqueous humor, right? That's this clear fluid-like runny, it's like water, right? That's all found within the anterior cavity. The posterior cavity is going to have vitreous humor. Vitreous, think viscous. Vitreous, viscous, right? It's a jelly-like thick substance that's going to help give the eye its structure and helps kind of keep it ballooned up, right? All right. So we've talked about the posterior cavity. Let's go ahead and talk about the different tunics of our eye. So first, we have the fibrous tunic. The fibrous tunic is just the kind of the whites of our eye, right? It consists of two different structures: the cornea, which is this clear film or this clear fiber that kind of covers over top of our iris and our pupil, right? Helps prevent things from falling into our eye. And then we also have the sclera, which is the whites of our eye, right? All of that around the cornea, or that clear portion. If I put the whole thing back together, you can see that whole sclera all the way around. And then that cornea that comes over top. All right, next we've got the vascular tunic. Vascular just means blood, right? So the vascularized layer of our eye is going to have blood in it. Let's go ahead and talk about some of those structures. So the first is the choroid. The choroid is just the layer on our eye. If you want to zoom in on here for me, if we look, there's the white layer right here, which is the fiber layer, the red layer, which is the vascular layer, and then this pink layer, which is actually going to be our neural layer, or our retina, right? And so the fibrous tunic is just this white sclera out here. Then we have the vascular tunic, this red layer right there. It consists of the choroid, which is the actual red tunic, or the red blood-filled layer within the eye, right? And it extends all the way up into the iris. The iris is actually a muscle. It's a sphincter muscle that helps to regulate how much light comes into our eye, right? And so it can dilate or it can constrict to allow for more or less light coming into the eye, right? And that's all the iris is. It's also part of that vascularized layer, right? So the choroid is everything behind it, beneath the retina, and then the iris is going to be that circular muscle that is kind of around the pupil, right? So that's this muscle right here. It's also that pigmented muscle, right? We know that the iris is very colorful and cool, right? All right. Next, we've got the pupil. The iris kind of forms a full loop around the front of the eye. If you want to kind of take a look there. This is the iris. It forms a full circle around the eye, and that circle opening in the center, that's the pupil. Everybody's heard of that, right? Nothing new there. All right, next, let's go ahead and talk about the sensory tunic. The sensory tunic, right? We've talked about the white layer, the sclera, the vascular layer, the choroid, and then now this tan layer, which is the retina or neural layer, right? This is the layer of our eye that actually captures light signals and helps to turn it into neural signals, right? And so this pigmented layer is all the way around the back of our posterior cavity, right? And it consists of the neural layer, which has photoreceptor cells. Photoreceptor. Remember, we talked about those at the beginning of the video. Photoreceptor. Photo means light, receptor means receiving, right? This is a cell that receives light. And so we have two different types of photoreceptors. We have rods and cones. Rods are we talked about these before, but remember, rods detect black and white light, whereas cones are going to detect color. All right, going back through here, we've got a couple specialized areas of the retina that you also need to be aware of. The first is the optic disc. So if we're looking at the retina all around the back of the eye, right, the optic disc is going to be found right here. It's where the optic nerve actually synapses in onto the eye, right? So we have the optic disc right there. It's an area with no rods or cones, and there's no light sensation that occurs there. It's the area of our eye that we anatomically describe as our blind spot, right? There's no neural sensation there. All it's doing is transporting signals back into that optic tract. Okay. Next, we've got the macula lutea. The macula lutea is going to be this little round structure right here. It's kind of small, but it's an area of greater density. It's an area that has a greater density of cones, right? So there's sharper vision acuity on this small little area right here. In the center of the macula lutea, we have the fovea centralis, which is the densest region of cones on the entire eye. That's where the lens is actually trying to focus light, right? That way it hits onto that fovea centralis and creates the clearest picture for us. That's why people also sometimes have to wear corrective lenses to help reorient how the light is hitting inside of their eye. That way it will direct light onto that fovea centralis, right? Super important structure to remember. All right, let's backtrack a little bit. So when light's coming into the eye, it's going to pass through the cornea, right? Passes through the cornea, passes through the iris, and it hits this structure right here. This is called the lens, and the lens sits right behind the iris and is suspended by a couple different structures that we need to talk about, right? The lens is actually held up by these suspensory ligaments, which if I take this part of the iris, right, you'll see how that it's almost like this underneath parts right here, right? The iris is going to be around the top of the, or around the anterior portion of the lens, whereas now we have those suspensory ligaments that kind of surround the entire lens and help to kind of hold it up. I'll even do this for you. Those white ligaments just hold onto the lens and they help suspend it in space, keeping it in front of that pupil. That way it can direct light where it needs to go, right? The suspensory ligaments are held onto by, so I'm gonna point out the suspensory ligaments quick. That's those right there, those white ligaments, right? They're connected to ciliary muscles, which are down beneath there. Those muscles actually hold on to the white ligaments or the suspensory ligaments that keep the lens in place. And they can bend and misshape the lens in order to direct light where it needs to go in the eye, right? Next, we have the ora serrata. So if we have the suspensory ligaments right here and the ciliary muscles down here, the ora serrata is just this serrated region kind of at the very back of those ciliary muscles, right? I use the word serrated like a serrated knife, right? A serrated knife is used to saw through something. That's exactly what this kind of looks like on an eye, right? It's just the back of those ciliary muscles kind of where they're connecting in onto the eye, and we call it the ora serrata. All right. Next, we're going to dive into the accessory structures of the eye. I'm going to go ahead and put it back together, and I'll see you in a sec. All right, so now we've got the accessory structures of the eye. First, we need to talk about the conjunctiva. The conjunctiva, if I take these accessory structures off, right, is going to be this pink layer just kind of on the surface of the eye, right? It's that pink layer, like if you take a look at my eyes, right, that pink, red, fleshy layer that kind of touches the white sclera on the eye, right? it helps to lubricate the eye and kind of, it provides mucus that keeps the eye able to move and rotate within its socket. The conjunctiva is very important for kind of lubrication and keeping the eye from getting too dry, right? And then there are a couple other structures that we need to talk about, like the lacrimal glands. All of these structures are really trying to keep the eye lubricated and also clean, right? We want to flush out any extra debris or particles that get in our eye that we don't want, right? And so the lacrimal glands are these glands found right up here. The lacrimal glands, if we looked on my face, right, would be found up here on the lateral side of my eye, right? That's the lacrimal glands. Lacrimal literally means tear, lacry, tear, right? Lacrimal gland is just the tear gland. This is the gland that actually provides that salty water that is used for our tear ducts, right? And so the lacrimal gland is going to secrete tears onto the surface of the eye, right, through the conjunctiva. And then those tears will travel along the surface of the eye, like washing stuff off or lubricating it until it travels down into a little hole at the bottom medial aspect of our eye called the puncta, right? I don't know if that's on this lab manual, but it might be in the future. So the puncta is that little hole right here that captures tears, right? The puncta is going to transport those tears into this like canal system, right? The canaliculi, the lacrimal canaliculi, that then transport those tears into the lacrimal sac. The lacrimal sac is this upper or superior portion of this kind of collecting duct that actually sits just above our nose, right? So this is just up here on our face, right? The lacrimal sac then drains down into the nasolacrimal duct, which we can't quite see, but that nasolacrimal duct is actually going to funnel down into our nose. Yes, how weird is that, right? Yeah, when you cry, have you ever noticed that you have to blow your nose a lot, right? Yeah, lots of snot starts coming out of your nose, right? And that's why, because the lacrimal glands release those tears, they travel across the bottom of the eye, they funnel into that puncta into the lacrimal sac up above here, right? Travel down through the nasolacrimal duct and enter in on our nose, right? And that's why you have to blow your nose when you cry. Kind of interesting, right? All right, let's go ahead and get the ear out so we can talk about sound and equilibrium. All right, so now we've got section 3, which is going to encompass the both section 3 and 4, which now encompasses the sound and equilibrium aspects of our ear. So first, let's talk about the associated cranial nerve, right? That associated cranial nerve is actually visible on this model, super helpful, right? And it's the vestibulocochlear nerve. It sits just back here and extends off of the spiral organ. We'll talk about that in just a second. So first, we have the external aspect of our ear, or the outer portion of our ear, right? And this is going to, the first structure that we need to know is the auricle. This is that fleshy mass, right? That part that has, or that fleshy mass of our ear that actually has like elastic fiber in it that can allow it to kind of bend and change shape and come back to its original shape, right? And we need it to stay that way because it's capturing sound and funneling it down into this tube, right? What is that tube? It's the external acoustic meatus, right? The external acoustic meatus is going to travel down through here. This is like this is kind of like that ear canal, right? Where it travels sounds and then funnels onto the tympanic membrane. The tympanic membrane is kind of this membrane, drum-like looking thing at the back of our ear canal, right? And it's going to actually capture vibrations and turn it into mechanical sound, right? It does that through the 3 different auditory ossicles, right? The auditory ossicles are 3 little bones that are unfortunately not on this model, but I'm sure you've seen them before. They're the 3 little bones that extend from the tympanic membrane all the way onto the oval window. The first one is the malleus, which would be right here where my pen's pointing. The second one would be the incus, which would be right here. And then the stapes, which is right up in here, that you can see where part of it's actually broken off, right? That's that stapes, looks like a stirrup bone, right? All right, next we've got the auditory tube, which is also known as the eustachian tube, that funnels down beneath the tympanic cavity right here, right? This whole area past the or behind the tympanic, or posterior to the tympanic membrane, is known as the
the tympanic cavity, right? Everything beneath that traveling down all the way into our throat, right? This tube goes all the way into our throat is known as the auditory or eustachian tube, right? And that tube helps us relieve ear pressure, right? Inner ear pressure. When we yawn or when we chew gum after we're on a plane, we can open the backs of that, right? Yep, our cameraman's doing that right now. And so you can hear the backs of your throat, or like the backs of this eustachian tubes kind of popping open and releasing pressure, especially when you're driving up to Laramie or if you're coming down from an airplane, right? All right. Next structures that we need to know are the oval window. The oval window is actually going to be right up in there. You can't see it. The bottom portion of this stapes muscles is covering it. But the oval window is actually the entrance into the inner ear or the cochlea, right? That's this whole structure that we're going to talk about is this vestibulocochlear apparatus, right? So we've got the oval window right here where sound enters, and then the round window, which we can't see on this model, but it allows sound to exit down through the auditory tube and kind of just flush out, right? Okay. Now let's talk about section 4. This is going to encompass the cochlea, right? So for the cochlea, we've got this smaller part of the model, and the cochlea is not this, not this stuff, right? These are the semicircular canals, and they actually help with balance. The cochlea is going to be this spiral organ, right? This snail-looking thing. That's the cochlea. First, we need to talk about the scala vestibule. The scala vestibule is going to be the top. There's 3 different cavities inside of this cochlea, right? There's the vestibule, or the scala vestibule, the scala media, and the scala tympani, right? That's that blue one. The scala vestibule is pink on this model. The scala media is purple on this model. And the scala tympani is blue on this model. And you can see just how those 3 different layers are kind of spiraling all the way up through this organ, right? And if you remember back to lecture, you'll know that there are hair-like projections that extend all the way through here that actually capture sound and turn it into the electrical signal that turns into what we kind of conceptualize sound as, right? Super cool. This whole thing is known as the spiral organ. Kind of easy to remember. It's spirals, right? I'm going to flip my lab manual here. And the last thing I want to talk about is the vestibulocochlear nerve. That's the nerve that extends off of the back of the spiral organ, and it, as you can see, connecting it with the larger model, it's going to extend off into the brain and up and into that auditory processing center, right? Okay, let's go ahead and talk about a couple more structures. Unfortunately, the models don't show these very well, but I still want to talk about them because I think that they're very important to remember. So, inner ear equilibrium. First, we have the vestibules, right? The vestibule is just going to be this area kind of between the cochlea. So that's this guy, right? Cochlea. Here's the vestibule, and then here's the semicircular canals. The vestibule is this region between the cochlea and the semicircular canals. It contains the utricle and the saccule, which is down inside here. Can't show it on this model, but the utricle and the saccule, if you look in your textbook, are going to help us recognize rotational balance, right? Or rotational movement. Next, we've got the semicircular canals, right? The semicircular canals are these things right here, pretty easy to remember. And they have 3 inner structures that I can't show you, but I can show you kind of where they're at, right? First, on the base of the semicircular canal is the crista ampullaris. That's this guy right here. The crista ampullaris is going to sense when this fluid brushes past a jelly-like substance, and that moves a hair that then registers that we've rotated our head in a right, left, or back and forward direction, right? And so we've also got the cupula, which is going to be inside of that crista ampullaris. That's that gel-like structure that holds the hair. And I counted 3, but there were only 2. We just needed to know the crista ampullaris and the cupula that's inside it. Remember that these are the semicircular canals connected to the vestibule, the spiral organ, and all of these signals are sent through the vestibulocochlear nerve, right? Sitting right here. All right, that is all of Lab 9, Special Senses. Good luck on your quiz this week, and I'll see you for the next one.